with Anna's talk with some examples of how you can go wrong. And so I'm going to follow up with Anna's talk with some examples of how you can go wrong. And so we've done lots and lots of experiments in the core, and we've seen lots and lots of people who have spent a lot of money and not gotten much out spending their money. So I want to reiterate what Anna said. If you're going to do metabolomics and you really haven't done it before, you should call the core, whoever you're going to work with, and discuss the experiment with them first off. We have been more and more turning down experiments simply because they are going to not work. Even though we like to take people's money, we don't like to take people's money and then have them mad at us. So these are some of the things I'll cover. These are obviously not exclusive. These are not exclusive to just one kind of experiment. I want to, one of the questions that you asked earlier about, translating stuff from tissue culture to animals. As Anna said, you can do it, but you have to be very careful about its time. Cell cultures, are very homogeneous. Animals are less homogeneous. The biggest problem is can you get enough signal for what you want to see and things like that. So, but we should talk. So the first thing I want to talk about is biological plausibility. This is probably one of the more important things that we talk to people about is how do you know that you're getting an answer that you actually can believe in? Because there has to be some biological plausibility to some experiments. Most of the time we're looking for metabolites that are associated with a specific phenotype, either another molecular phenotype such as gene transcription or a clinical phenotype. And so you need to have a range of phenotypes to be able to understand what these relationships are, especially if you're doing higher throughput untargeted. This is one I like to call the biological plausibility that metabolomics responsible seen. This is an exposure experiment. There's exposure 1, phenotype 1, exposure 2, phenotype 2, and this is a set of samples that an individual brought to us, and we actually ran the metabolite data, which is the phenotype, before he actually got all the results back on the exposure data. And the exposure data, the bottom line of this, so here is the presumptive limited detection, although the assay could get lower levels. But just about every quote exposure is lower than what can be accurately measured. And so if you do the phenotypes before you actually do if you do your molecular phenotypes before you actually have the data that you're going to correlate it with, you can be in trouble. So don't presuppose that you're going to get the data that you think you're going to get, because these two people here really weren't associated with any sort of molecular phenotype. This one person that was up here, obviously there was not. And if you don't have a range of exposures, it's going to be really tough to do. So these are the kinds of things, if you call us up, and so do you have the data in hand for what you're going to do instead of doing it in parallel? Instead of spending $20,000 in the lab, it's probably better to make sure that you have what you think you have. Extraneous variation in phenotypic endpoints muddle relationship to the metabolome. So this is a lipid, this is from a paper that was looking at the fatty acid profiles and the ratios between intervention, a drug treatment for fatty acids, and assess the change in fatty acids before and after a statin intervention. They're positing that the statin that they were testing had special properties because it changed the fatty acid intervention. And so they treated a number of different people. And what it turns out, when you actually started reading their proposal closely, is that the people who have these higher levels of polyunsaturated fatty acids instead of getting a statin, they were given fish oil. Now, fish oil is polyunsaturated fatty acids. And so their claim that there was changes in this because of the drug was actually because of a drug, but it was actually not the drug they were thinking of. And so being very careful because, metabolomics is a very sensitive thing. And as Anna pointed out, diet is really, really incredibly important thing. I showed you in the genomic slide, genetics has an impact, but on top of that, really diet is probably the most important thing that causes a variation in the metabolome. And so trying to keep that stable. I'll show you tomorrow, I'm going to show you some data that shows that the metabolome can actually change very, very quickly. We've done some metabolomics after one meal, a mixed meal tolerance test, and things dynamically change very quickly. And if you feed even for 1 or 2 days a specific diet higher in one nutrient than another, you can actually see a very rapid change in the metabolome. So really what people eat in the last couple of days can make a big difference in where you're starting from and the diversity in the metabolome that you have. So diet is really important. So for many of our experiments that we do now, we actually ask people, we give them a diet that is pretty much 50 percentile of the human or American diet, 50 percentile for the macronutrients at least, and to try and get everybody to come together a little bit more. The genetic variation is still there. But sometimes this actually will get rid of a lot of variation that you're able to pick up a signal in your data a little bit easier. So it's one of the things to remember that what you eat is really important. This is another one that's actually, this is a paper that was in Science Translational Medicine. It got a lot of press that suggested that there was differential metabolic effects of gastric bypass versus dietary interventions in obese diabetic subjects despite identical weight loss. Okay? So when I read this paper, and I'm sort of bummed because we actually were doing these studies ourselves, trying to compare the differences between bariatric surgery, and we didn't see as big a change as they saw. That we saw that diet actually had in some of these metabolites branched chain amino acids, which was their primary endpoint of this. The branched chain amino acids changed more with gastric bypass surgery than they changed after weight loss, identical weight loss with medical intervention. And we were confused about this. And so, because we had looked at this, and so we actually had done a study where we took people, changes in amino acid levels while in a normal or a very low calorie diet. And so we had people lose 15% of their body weight, and here's the p-value for these different amino acids, and the branched chain amino acids are here as valine, leucine, isoleucine are over here. And you can see that on a normal diet, we actually had statistical differences in these, but on this very low calorie diet, while they were on the diet, they didn't have a difference. And so this was somewhat confusing to us, or not confusing, but was interesting because when we went back and looked at the paper that they referenced for the original diet population, that these people were kept on their 1,000 kcal diet, which is a very high protein, very low calorie diet. And so we used basically the identical diet, and when we did it, when you're on this diet, like they were there, you don't see a statistical difference between the branched chain amino acids, because these diets, actually 27% of all the amino acids that are coming into the person are branched chain amino acids. So very little things can make a difference on how you might be able to interpret your data. So it's really important, again, to think about what people are eating, what's their state of nutrition when they do it. Anna talked about feed-fast cycles, and it is really important to try and get everything to be as consistent as possible. It'll reduce noise, and it'll reduce what I believe was an erroneous statement by this group. This is the most common thing that we get, and we try and get people not to do these kinds of experiments. This is early on. These investigators sampled men and women with and without polycystic ovary disease, with and without obstructive sleep apnea, during slow sleep and fasting in the morning. All right. So now you take, the 80 people here and you divide them into this many different groups, you know, a PSLDA plot of untargeted metabolomics data, you can separate just about anything on PLSDA. But if you do a PCA plot, these things just don't separate. And really, it's having too many variables at once can just kill you. You know, we worked with them trying to collapse these down into smaller groups. The problem was that the slow wave sleep, they were feeding people before they went to bed, or giving them actually about 20% of their calories before they went to bed to try and get everybody the same. They ate the same thing, but the slow wave sleep was happening about 4 hours before the fasting in the morning, and so therefore there was a difference even in there. And so it was really difficult to do. Sex makes a difference to your metabolome. So men tend to have higher amino acids in their blood because they have a higher muscle mass. And so when they fast and you mobilize amino acids for gluconeogenesis, when you're fasting, you're going to have higher levels in men. And so when you start, so sex makes a difference. Age makes a huge difference in your metabolome. So the age of somebody, for most of the common metabolites that we measure, I showed you in the slide earlier today that there's a change in the metabolome. So sex, age, BMI, the last study that I just showed you, there's a huge difference between thin people and obese people and what their metabolite levels are. Insulin resistance, all those kind of things. So you need to think about all these things, especially when you do your statistical analysis, you need to take these things into account. Now I know you all have a sense of this, but you know, taking a look at your data and just dropping lines over the metabolites can really give you some information about which are the things that are actually making a difference for the metabolite levels. There are racial differences, especially in the lipidome, that we've identified between individuals, and so you need to take that into account as well. And then diet. So different, you know, if you had the Asian people in my laboratory versus the Westerners in my laboratory, they clearly eat a different diet because I sit right across from the microwave. And sometimes it's really smelling good, and sometimes I don't understand. It's really, other thing is really important is labeling and timing. Labeling all the time, and Tanu actually, you know, we had this interesting data set that somebody sent us, ran about 100 samples, and investigator presided these samples for lipidomic profiling, saying there's a subset submitted samples where some subjects sampled twice about 10 years apart. And so this is our data form that we asked people to bring in. And when we actually looked at the age differences with the same subject ID, what we saw is that there was between what they stated on the enrollment form versus what the difference was the age that they stated they were versus the age because of the birth dates, or the birth dates and the time collected. There's quite a huge variation in the age of individuals. Now, if you're going to try and adjust for age and you don't know what the age of the individuals are, it's really hard to help. So it's really important to really, and Anna said this too, and I'm trying to reinforce everything she said, is that you need to be very meticulous about time of day. And she showed you some circadian rhythm stuff. I think she had that in her talk. You know, time of day, fasting, all the different demographics that you can, because you know metabolites change fast, and little things can make a big difference. So it's really the upstream, making sure that you have things right and you make sure you have things labeled correctly. Now, it turns out that you know, the samples were actually really interesting anyways, and these are, they're showing you that time does make a difference, especially if you're doing retrospective studies. This is from my friend Craig Hannes. This is a small study that we did looking at samples from 1985 to 87, samples collected during these years when the prevalence of type 2 diabetes in Starr County, Texas, the poorest county in the United States, the prevalence of diabetes was about 13-14% in 85, 87, and about 45% only a couple decades later. And you can see this is a correlation heat map of some isocarotene amino acids, fatty acids, triglycerides, and different phospholipids. And we'll talk about this, I think, tomorrow when I talk about correlations. But you can see that there's a difference in the pattern of the correlation, and that these samples here have much higher correlations among these metabolites. And I'll talk about this, but what we actually found doing this study is that, one, that the triglyceride clinically measured, and the triglyceride when we add up the triglycerides from all the measurements, we actually had a really good correlation in all the samples, such that even after these triglycerides were clinically measured back in 1985, and when we actually did it, and this was done a couple of years ago, so about 30 years later in some cases, there was really a great preservation of a lot of different metabolites. If we looked at leucine levels in C5 acylcarnitines, and acylcarnitines are very stable compounds, that these are some of the things that you can measure in blood spots for inborn area metabolism. And they're used because they're very stable. Leucine generates acylcarnitines, and there's usually a very high correlation. What we found, even in these old samples, leucine is good. Some of the things, Anna showed you that some of the amino acids break down more quickly than the others. Aspartate and glutamate are two metabolites. If you see differences because of collection or sample storage, you probably should ignore some of those amino acids because they do break down. The biaminated amino acids, that one of the amine groups can come off, and you actually get more glutamate from the glutamine, and aspartate from asparagine, deaminated asparagine, ornithine can come up. So they're there. We think this actually, and I'll talk about this, that this greater correlation is actually due to the higher intake of carbohydrates. There's ways that we can actually see this in the metabolome by looking at these correlations. And it's because you're just shoving a lot of saturated and monounsaturated fatty acids into the system, and everything sort of fills up with those, and they become more highly correlated. But I'll come back to that. So you can take samples over time, but the incidence of diabetes in this population is directly related to one thing, which is the number of fast food restaurants in the county, which probably is reflected in the metabolome. So if you're actually doing retrospective samples, you have to make sure that you understand exactly what's going on in the environment of these individuals. All right. Extraneous factors affecting metabotypes. So this was an interesting experiment. We learned a lot from this, especially how to talk to people very closely about how to do their experiments. This is a cell culture experiment looking at fluxomics, and you'll learn about fluxomics where you put a heavy isotope in with your samples, and you can actually watch the carbons flux through. And so these were assessed for static levels of metabolites and then fluxomics by addition of uniformly labeled C13 glucose for 0 to 30 minutes. The pre-flux experiments were under two different conditions. In A, the cells were fed 24 hours previously, washed and harvested at time 0, here. And then at T, 30 minutes after addition, or at T equals 30 minutes after addition of 10 millimolar UC13 glucose. So here. So you can see B cells were fed 24 hours previously, washed and refed for 30 minutes, and harvested at T and 30. So they treated these cultures a little bit differently. These cells were 24 hours and then spiked in to add these the high labeled glucose, where these were freshly fed and then spiked in to add the glucose. Now in the end, these look pretty much the same, but the baselines were much different. And the reason the baselines are different is because these cells are starved. Basically, after 24 hours, highly metabolically active cells are depleting a lot of the nutrients in their media, and therefore you get to a different thing. It's sort of like feeding the same person the same diet. You need to feed the cells the same diet before you start doing your experiment. So most of the times we suggest that people refeed their cells with fresh media for a short amount of time, and then take that media out, then put in their experimental media if you're going to do it. And so you have to think about what you're comparing what to, and it can make a big difference as far as the levels of metabolites that you have. One of the things that we've actually been successful at doing is figuring out this labeling time. I don't know if Charles is going to talk about seahorses and all that kind of stuff. So in flux experiments, one of the things is that you need to have enough time to label. And we've actually been working with the Agilent people, Agilent now owns Seahorse, talking to them, but we now routinely ask if people have put their cells, their cultured cells, in a Seahorse apparatus, because you get an idea of what the oxygen consumption rate is, really gives you a good idea of how much metabolism, how fast metabolism is, and how long you actually need to label your cells. So if you have some idea of that, or before you do a flux experiment, it's actually a really good thing to understand, because some cells are really slow and some cells are really fast, and how long you label is going to make a difference. So we tried to understand, two people sent us two different sets of macrophages. This set where we did, again, these labeling experiments, there was a lot of C2 citrate being made. It was very, after 30 minutes, there was about 50%, or nearly 50% was labeled in the two different treatments. And these people only had had much less labeling. And we started talking to the individuals. Okay, so exactly how did you do your experiment? Because we sent them this experiment, the results of this, and this is what you should expect. And they were really disappointed when they got this. And so, so we talked and talked and talked, and finally we found out that these people actually added a little bit of interferon to the cultures. That was their standard protocol. These people didn't, and it actually turns out to be a really thing. Sometimes it's really fun to be a detective to try and figure out exactly what's going on. You can, this is mistaken interpretation of data. So this is an experiment that we did. I showed you everybody else's mistakes, and now I'll show you our mistake. Well, this was a study we did looking at beta cell metabolism and looking at the amount of citric acid, glutamic acid, malate, and aspartic acid that we can see in these cultures during a time course. And so I'm sure you all remember your Tca cycle. So citrate is here. Glutamic acid is in equilibrium with alpha-ketoglutarate. Aspartate comes from the outside and feeds into the Tca cycle, into oxaloacetate through this malate aspartate shuttle, and malate is actually at the end here. So we said, oh, well, this is pretty cool. The citrate is in pretty high concentration, but look at the malic acids in pretty high concentration as well. And what you see is there's a marked increase, and we just assumed it was coming from the Tca cycle. But when we actually did a flux experiment, we added label to it. What we found is that if you were adding labeled glucose and labeled pyruvate, which will go labeled citrate, what you would see is that if this malate is coming from the Tca cycle and you get this marked increase, you should have a lot of label. What we found is that a lot of it was unlabeled. So most of it was unlabeled, actually. And so through some other experiments, what we found is that all this malate is actually coming from aspartate that's going to oxaloacetate, which is actually going backwards into the malate. And so you're actually getting a lot of malate accumulating in the cell simply because of that. So sometimes, especially when these guys know it from the cancer cells, you really need to a lot of times do labeling experiments to make sure that you're interpreting where your different metabolism is coming. I just rejected a paper that was into a very prestigious journal simply because the people refused to do a flux experiment. They just wanted to use metabolite levels and making a claim that they couldn't support. And actually, part of my insistence is because we've actually done a similar experiment for somebody else, not for these people. And it's hard to write in a review going, I know this is wrong because we've done this before, and so it's really tough. And so, I wrote to the editor as a thing, and they rejected the paper simply because there's a lot of times you really need flux to be able to really understand where things are coming from. Okay. And then this is the other detective one. This is with Tanu, and it took a while, and actually our colleague who submitted this sample. So this was a bunch of samples that he had collected from a human study. And they were so they're collecting the samples, and they were getting DNA from the samples as well. And so submitted it. We did the lipidomics platform on it, and what we found is that that when you did this, we found is a subset of the individuals here we're not grouping with the other set. So this is, in here was our test pool, black. So you can see after normalization and all that kind of stuff, you have a test pool that's here, and usually what we see is the metabolites are scattered out in a PCA plot around the test pool, which is where we add a little bit of each sample into a pool and then run those, and that usually should be in sort of the middle of this cloud that you get. But these samples here, which were part of this test pool, are sitting out in left field here, and no matter what you did, you couldn't figure it out. And when we looked at the lipids that were associated with this, they were all saturated fatty acids containing lipids, so triglycerides and phosphatidylcholines that were associated with fatty acids. And it was really difficult for us to understand. And so so these were all samples were collected in the clinical research unit. And it turns out what happened here is that somebody else was collecting the samples or separating the samples. So they would deliver the blood fresh from the patient, they would spin it down, and here what they would do is that they would take the plasma off and aliquot out the plasma so we could actually do this. But this person actually thought that they had learned it the right way, and they went into the plasma, took the white blood cell, the buffy coat, the white blood cells, and pulled it out through the plasma. And that difference caused that change. And the way that Simon figured it out was that he actually did cytokine assays on the plasma and found that this plasma was much higher, had much higher concentrations of cytokines in the plasma. And then when he went back and actually then figured out that it must have been some sort of cellular contamination, he actually figured out how this was. Now, why was only saturated fatty acids coming out? I'm sure we still don't quite understand that. Well, it may be because there's maybe more membranes or some of the cells were bursting and some of the triglycerides were coming out. I don't know. But it was clear that there was a difference. And so this is these little things. So it took about a month to figure out why this was. And really, so this data is really much less useful than this data because the sampling was done incorrectly. So if you're thinking about taking DNA, make sure you take the plasma first. Don't stick something in and drag something through your plasma because it's going to contaminate. All right, so be careful in the design of your studies, fast-fed, who they are, what their sex, what their weight, all those kinds of things. Metabolome changes quickly, and it's clearly influenced by the nutritive status of both the people, the cells, the animals, all these kinds of things. Really makes a difference. One of the things that we find makes a big difference, if you fast a mouse for 4 hours, or some people like fasting mice overnight, if your eye or your UKUCA will let you do it. So it's actually frowned on these days because remember, mouse metabolism is 15 times faster than yours is. And so if you fast for 12 hours a mouse, it's actually like fasting for 3 days. They lose 15% of body weight. And their metabolome changes a lot from a 4-hour fast to an overnight fast. And really is a big difference. And there's a very rapid decline in a lot of metabolites in that. And so depending upon how long you fast, if you're an hour later, hour earlier, your metabolome can change in animals that have really high metabolic rates. So just another thing to remember. Target on-target metabolomics gives you snapshots in time of sampling. It's not telling you where the data came from. And check and recheck your submitted samples. Don't waste your money. We've learned that people can get upset with us if they don't get the results that they think they should get. So we're much more buyer beware with a lot of our customers these days. So I'm going to stop there, and if there's anybody with some questions. Yes.
Just like in all types of omics studies, there's always non-specific background. And I'm wondering, is that all going to be weeded out in the initial threshold dropouts? Or is there a kind of published work that we could kind of look at and see if there's anything in our analyses that probably shouldn't trust?
So in other words, should you see this metabolite, we don't see it, or we see a metabolite that shouldn't be there. It's really interesting. So I'll use alpha-ketoglutarate or as one of the examples. Some cell lines you can see a lot of alpha-ketoglutarate. Some cell lines you can see a lot of acetyl-CoA. Some cell lines, and especially in tissue, acetyl-CoA is actually incredibly hard sometimes to see because it's actually just being consumed very quickly. And so I'm not sure there's actually a great reference. I mean, for plasma, there's published values of different metabolites. In cells and culture, especially the funny cells you guys work with, I don't think there's a steadfast rule of what you can find and what you can't find. It also depends on how people do it. So LC, liquid chromatography mass spec doesn't see alpha-ketoglutarate as well as the GC mass spec, and so we'll run some of those. It's hard to see lactate sometimes, good lactate peaks on one platform versus another. So if you're interested in a specific metabolite, it's really important to talk to the spectroscopists about it, about what you really want to measure and what you really want to see. Now, when you're doing these untargeted things and you're just looking to see what's there, all bets are off. Well, not all bets are off, but a lot of bets are off because everything is a little bit different. It depends on how you do the experiment, under what conditions, how you feed them, how old the cultures are. Are they confluent? Are they not confluent? That's another thing, that confluences makes a difference. 3D culture, you get a different answer than you do in 2D cultures. And so these are all things that you all sort of have a understanding of, but metabolism is lots faster than mRNA changes. It's even faster in phosphorylation, dephosphorylation as far as total changes that you see. And so remember, it's fast, especially cell culture, is fast and reproducible. And we have a lot of protocols that we've worked out to try and make this a little bit better. I think Charles is going to talk about animal work, but we can send you a reference. Matt Lorenz actually, for the one paper that I showed up there where we had a mistake, he spent a lot of time and has published an analytical chemistry paper on how to stop and extract metabolites out of different cultures to try and get the least variability and the best recovery that you can. Dr. Banerjee.
You know me.
Yeah.
So
intimately.
So
well, we know your samples.
Yeah, my samples are a pain. But speaking of, so when you are estimating the level of metabolites, it doesn't, or does it necessarily mean the absolute amount present, or will you also factor in the enzyme activity? So for example, like not finding something can also mean that the next enzyme is like super active.
Yeah. So the biological interpretations of we can measure what you give us. The biological interpretation is up to you. We'll help, as you know. We like looking up papers and trying to figure out what's going on, but the people get mad at me for sometimes not suggesting to do the right experiment or whatever. I get mad at people for doing a large experiment, then coming to me with a data set and saying, so now what does this mean? It's sort of like, well, I am not a microbiome expert. You have to tell me what it means. We try, I learn because I like reading literature and I try and find things. But, it really is incumbent upon you to interpret your data in the context of the biology that you're doing. I don't know stem cells. You know stem cells. And so, but there's always somebody that's done something that's close to what you've done, and so at least it'll give you a guide. I mean, you know what I'm talking about. Looking in the literature, there's a lot of stuff going on. There's actually some very good blogs. I forget the one that Kostas sends out. He sends out a list of papers. So, but Metabolomic Society, they send out a blog with a list of a lot of interesting papers that you might be interested in. New techniques and surprising findings and technologies to help you do this kind of thing. And so I'll get a couple of them. We can share some of those links with you to so you can actually get more and more of these things. Any other questions? All right.
There are no more questions. So one quick thing: if you park in a patient parking, you have been informed that you cannot do that, and you get